In the presence of oxygen, the way that a cell produces ATP molecules that are used as the energy source in that cell is via certain types of processes that together are known as aerobic cellular respiration. And this includes glycolysis, pyruvate decarboxylation, the citric acid cycle, as well as the electron transport chain. Now, glycolysis breaks down our glucose into 2 pyruvate molecules as well as a net of 2 ATP molecules. And in the presence of oxygen, those 2 pyruvate molecules will then go into the mitochondrial matrix, where they will undergo a series of processes that eventually produce NADH molecules. And those NADH molecules then go on to the electron transport chain, give off their electrons onto the enzymes of the electron transport chain, and the NADH molecules are oxidized back into the NAD plus molecules. And these NAD plus molecules can then be reused in the process of glycolysis, pyruvate decarboxylation, and the citric acid cycle. So we see that under aerobic conditions, basically the electron transport chain is where we synthesize the majority of the ATP molecules, and it's also the location where we oxidize the NADH back into the NAD+. Now, the question is, why do we have to actually regenerate these NAD plus molecules? Well, basically because without NAD plus molecules, glycolysis will not take place. So things are pretty simple during the presence of oxygen when we have enough oxygen present in the cell. But what happens if we do not have any oxygen present in the cell? What happens if we do not have enough oxygen present in the cell? So these types of conditions, when we do not have oxygen, are known as anaerobic conditions. And under anaerobic conditions, glycolysis is the only method by which we actually produce ATP. Remember, glycolysis can take place regardless of whether or not we actually have oxygen present in the cell. So glycolysis is the process, the first process of aerobic respiration, and it's the first process of anaerobic respiration as well. However, under anaerobic conditions, glycolysis is the only method by which we produce ATP molecules in the cell. Now, the problem with this is glycolysis uses up NAD plus molecules. But in order for glycolysis to continually take place, we have to somehow continually regenerate the NAD plus molecules from the NADH molecules. And the way that we regenerate these NAD plus molecules that are needed by glycolysis is by the process of fermentation. So once again, under anaerobic conditions, glycolysis still occurs, which means that we break down glucose into 2 pyruvate molecules and produce a net of 2 ATP molecules. In the process, we also deplete our pool, our concentration of NAD plus molecules. But we need these NAD plus molecules for glycolysis to continually take place. So that implies that we must have some way to regenerate the NAD plus molecules by oxidizing NADH back to NAD plus. And since under anaerobic conditions, the NADH molecules do not actually go onto the electron transport chain, another method of regenerating the NAD plus molecules is via the process of fermentation. And there are 2 types of fermentation processes. So we have alcohol fermentation and lactic acid fermentation. So let's begin with alcohol fermentation. So certain types of bacterial cells as well as yeast cells basically undergo this process of alcohol fermentation. So glycolysis is the method by which they create ATP molecules, and in order to continually create those ATP molecules, our yeast cells as well as bacterial cells have to actually regenerate the NAD plus molecules. So in this type of fermentation, basically pyruvate is first, or pyruvate first undergoes the process of decarboxylation to produce acetaldehyde as well as the carbon dioxide. And then our acetaldehyde reacts with our NADH and an extra H ion to form our ethanol alcohol as well as our oxidized NAD+. So this is exactly why we call it alcohol fermentation, because we form ethanol. In fact, this is the method by which we actually synthesize alcohol that is then sold in bars and so forth. So we see that alcohol fermentation involves the process of glycolysis as well as this process here. So in yeast cells, for example, they produce ATP, and to continually produce the ATP, the pyruvate basically undergoes these two steps to produce ethanol as well as regenerate the NAD+, that can then go into our glycolysis cycle to produce even more ATP molecules. So this is how we actually regenerate and we reuse our NAD plus molecules. Now let's move on to a more common cycle that takes place in human cells, known as the lactic acid fermentation. So in certain cells, including human cells, bacterial cells, as well as fungi cells, when oxygen supply runs low, for example, during some type of vigorous exercise, too many pyruvate molecules and too many NADH molecules are produced. And because not all pyruvates and NADH molecules can be placed through the citric acid cycle and the electron transport chain at the same exact time, we have a buildup of NADH molecules that takes place. And so we have a very little amount of NAD plus present in the cell, and we need NAD plus for glycolysis to take place. So what the cell actually does is it undergoes this lactic acid fermentation process, in which a special enzyme known as lactate dehydrogenase transforms our pyruvate. It reduces the pyruvate into lactic acid, as well as forming our oxidized version of NADH, our NAD+. And this NAD+ can then be placed into the glycolysis cycle to produce our continual source of ATP. Now, lactic acid can basically break down into its conjugate base, lactate, and this releases H+ ions into the cell, and that means it increases it decreases the pH of the cell by increasing the concentration of H+ ion. So the buildup of lactic acid in the cell decreases its pH, and it's the decrease in pH that basically causes the feeling of fatigue following our vigorous exercise. Now, the lactic acid must eventually be transformed back into pyruvate, and this takes place in the liver. So once we form our lactic acid, the lactic acid travels back into the liver, and inside the liver, the lactic acid undergoes a process known as the Cori cycle. In the Cori cycle, we basically use oxygen to transform our lactic acid back into pyruvate, and the quantity of oxygen that we have to use to transform the lactic acid back into pyruvate is known as the oxygen debt. So we see that the process of fermentation is the method by which we basically regenerate our NAD plus molecules from the NADH molecules, because we need our NAD plus molecules to basically undergo the process of glycolysis and produce our continual source of ATP. And our fermentation only takes place under anaerobic conditions. Remember, under aerobic conditions, we have our citric acid cycle and we have the electron transport chain that actually produces the majority of the ATP molecules as well as oxidizes the NADH back into the NAD+. But under anaerobic conditions, we have glycolysis being the only method by which we produce our ATP molecules, and to regenerate those NAD+, we have to undergo the process of fermentation. Now, whenever somebody says fermentation, that basically incorporates glycolysis. So glycolysis followed by this process or this process together is known as fermentation. So fermentation produces a net of 2 ATP molecules when we take one glucose and break it down. So we see that these processes do not actually themselves produce any ATP molecules. It's only glycolysis that produces those ATP molecules. And when we break down one glucose molecule, we always produce a net of 2 ATP molecules. So if this cycle takes place once, we produce a net of 2 ATP molecules.